Welcome everyone to another episode of Vet Neuro Jar, the podcast where we explore the fascinating world of veterinary neurology. I'm your host, and today we're diving deep into a topic that's absolutely critical for understanding brain health, not just in our furry friends, but in ourselves as well, the blood-brain barrier. You may have heard of it before, but what exactly is the blood-brain barrier? Well, it's essentially a super-selective security system for the brain. Imagine a microscopic fortress surrounding the brain, carefully controlling what gets in and what stays out. It's a specialized network of blood vessels that separates circulating blood from the brain's delicate extracellular fluid. This barrier is crucial for maintaining a stable environment for the brain to function properly. Think of it like this: your brain is a highly sophisticated computer, and the blood-brain barrier is like a surge protector and a dust filter all in one. It ensures that only the right nutrients, oxygen, and essential compounds can enter. At the same time, it blocks toxins, pathogens, and other harmful substances that could cause damage. It also makes sure waste products from brain metabolism are moved out. This is vital for keeping brain pH levels steady and preventing plaque formation associated with aging and neurodegeneration. This barrier isn't just some passive wall. It's an active and dynamic structure. It's made of several cell types working together to maintain this delicate balance. These cells include specialized brain endothelial cells, which line the blood vessels. These endothelial cells are very different from those found in the rest of the body. They have unique properties that make them highly selective in what they allow to pass through. These specialized brain endothelial cells are knit together by tight junctions, which act like incredibly strong seals between cells. These tight junctions prevent materials from simply passing between cells. Instead, materials must go through the cells. This makes the barrier highly effective at controlling what gets into the brain. It's like a customs checkpoint where everything is carefully inspected. This cellular protective mechanism is acquired early in life. Another vital component of the blood-brain barrier is the presence of astrocytes, which are star-shaped brain cells. Astrocytes have foot processes that wrap around the blood vessels. These foot processes not only help provide structural support, but also help control blood flow and the movement of nutrients across the barrier. Astrocytes are involved in the transport of nutrients, such as glucose, across the barrier. Glucose is the primary energy source for the brain. Astrocytes deliver glucose or energy substrates to neurons. These foot processes also help to regulate water balance in the brain using special water channels called aquaporin 4. Astrocytes can also regulate the function of tight junctions and a key efflux transporter called P-glycoprotein. Finally, parasites are another cell type that wraps around the blood vessels and plays a critical role in supporting and regulating the function of the barrier. Parasites interact with brain endothelial cells and astrocytes to provide structural support and control permeability. They also help with the development and maturation of blood vessels in the brain. Dysfunction of parasites is linked to several brain disorders. Now, this might seem complicated, but all these components work in harmony to keep the brain healthy. The structure of the canine blood-brain barrier is similar to that of humans, making dogs a valuable model for studying this complex system. Also, like humans, dogs are prone to neurological conditions, such as epilepsy, cognitive dysfunction, and brain tumors, which are very similar to those seen in humans. This means that what we learn from studying the canine blood-brain barrier can potentially benefit both species. One of the most important ways that the blood-brain barrier protects the brain is through a special type of protein called P-glycoprotein. P-glycoprotein belongs to a family of proteins called ADP-binding cassette transporters, and it acts as an active efflux pump on the surface of the brain endothelial cells. Think of it like a tiny bouncer at the door of the brain, actively pushing out unwanted substances, including many drugs, and keeping them from accumulating in the brain. This transporter is particularly important because it limits the penetration of potentially harmful substances, known as xenobiotics, into the brain. However, there are situations where the effectiveness of this protection is reduced, which is a problem. Specifically, a mutation in the gene that encodes P-glycoprotein, called ABCB1 or MDR1, is common in some dog breeds, especially collies, Australian shepherds, and some other herding breeds. This mutation can result in a less effective version of P-glycoprotein, or even a complete lack of P-glycoprotein activity. If the bouncer at the door is not doing its job, then the brain becomes more vulnerable to harmful substances. For example, the drug ivermectin, which is commonly used to treat parasites, can become very toxic to the brain when P-glycoprotein isn't working properly. This is why some dogs have severe reactions to ivermectin that can be life-threatening. In contrast, other similar drugs, like selamectin, are safer for dogs with this mutation because other, as yet unknown, transporters may be at work. There are a number of other drugs that are affected by this mutation, and some common examples include the tranquilizer acepromazine, the antiemetic apomorphine, and the antitussive butorphanol. For dogs with the mutation, these medications might require a significant dose reduction to avoid toxicity. For other drugs like the chemotherapeutic agent doxorubicin and the anti-inflammatory grapaprint, doses should be significantly reduced in dogs with the mutation. Some medications such as the antidiarrheal loperamide should not be given at all to dogs with the mutation. It's also important to note that the clinical signs of ivermectin toxicity in dogs are well documented and can include neurological and ocular manifestations. These can range from depression, lethargy, and tremors to seizures and even coma. In some cases, dogs can even experience blindness. Interestingly, P-glycoprotein is not just important in the brain. It is also found in other barrier tissues in the body, such as the blood-arachnoid barrier, blood-spinal cord barrier, biliary system, intestines, placenta, yolk sac, and renal tubules. So, if there is a problem with P-glycoprotein in the brain, it may also mean that P-glycoprotein is not working as it should in these other tissues. P-glycoprotein function can also be impacted by other factors, not just gene mutations. For example, infections and inflammation can disrupt P-glycoprotein activity. This means that during an infection, the brain may be more exposed to harmful substances. Exposure to some environmental toxins may also affect P-glycoprotein function. Also, some medications can interfere with P-glycoprotein, which is an important consideration for veterinarians to be aware of. This is a reminder that the blood-brain barrier, while normally protective, is a very dynamic system and its function is affected by many factors. The blood-brain barrier can also be disrupted by various diseases and conditions. For instance, inflammatory conditions like meningitis and encephalitis can damage the blood-brain barrier. These conditions can cause immune cells and inflammatory molecules to enter the brain, leading to increased permeability and further neurological problems. Sepsis, a severe systemic inflammatory response, can also damage the blood-brain barrier. Even steroid-responsive meningitis arteritis, an inflammatory disease that primarily affects the central nervous system, involves disruption of the blood-brain barrier. Infections from viruses, bacteria, and fungi can also lead to inflammation and compromise the integrity of the blood-brain barrier. Cryptococcus fungi, for example, can enter the brain using a Trojan horse mechanism by using infected macrophages, or by passing between damaged brain endothelial cells, or by transcytosis. Some pathogens can reduce P-glycoprotein activity, increasing the risk of toxin exposure in the brain. Canine distemper virus is also known to cause inflammation and neurological issues. In addition, systemic bacterial infections can cause significant neuropathologies including meningoencephalitis. Even protozoan parasites like leishmania can disrupt the blood-brain barrier. Aging is another factor that can compromise the blood-brain barrier. As dogs age, the barrier can become more permeable. This age-related dysfunction has been linked to cognitive decline, similar to Alzheimer's disease in humans. It's possible that P-glycoprotein activity is reduced in aging dogs, contributing to cognitive decline and neurodegeneration. This is something that scientists are still trying to fully understand. Traumatic brain injury is also a major cause of blood-brain barrier disruption. Physical damage to blood vessels can lead to leakage of blood into the brain. This can cause a cascade of secondary injuries that includes inflammation, swelling, and oxidative stress. Also, inflammation and hypoxia after traumatic brain injury may further reduce P-glycoprotein activity. Even brain tumors can disrupt the blood-brain barrier and lead to brain swelling. This can also change how drugs reach the brain and influence their effectiveness. Epilepsy is yet another condition where the blood-brain barrier can be disrupted. Research has found that disruption of the blood-brain barrier can cause seizures, and in turn, seizures can lead to further disruption of the barrier. This creates a vicious cycle that can make epilepsy more difficult to manage. Interestingly, some studies have shown that in certain cases, dogs with the mutated ABCB1 gene and epilepsy may actually have a better response to phenobarbital, a common antiepileptic drug. This is potentially due to the fact that reduced P-glycoprotein activity allows more of the drug to enter the brain. It's also important to know that the expression of another enzyme called cyclooxygenase 2 is increased in epilepsy, leading to increased levels of prostaglandin E2, which may actually increase P-glycoprotein expression and limit the effectiveness of anti-seizure medications. So, how do veterinarians evaluate the integrity of the blood-brain barrier in dogs? There are a variety of methods, including imaging techniques like dynamic contrast-enhanced magnetic resonance imaging and computed tomography. These techniques use contrast agents that are normally kept out of the brain by the barrier, but if the barrier is disrupted, these agents can leak into the brain tissue and be visualized using imaging. There are also laboratory tests, such as cerebrospinal fluid analysis, that can show if there is increased protein in the brain fluid. These tests all provide important insights into the functionality of the blood-brain barrier. It's becoming increasingly apparent that the study of the canine blood-brain barrier has significant implications not just for dogs, but also for human medicine. Many of the neurological conditions seen in dogs have parallels in humans, such as Alzheimer's disease, multiple sclerosis, Parkinson's disease, and traumatic brain injury. In Alzheimer's disease, for example, blood-brain barrier dysfunction is linked to impaired clearance of amyloid beta protein from the brain. Similarly, in Parkinson's disease, disruption of the blood-brain barrier can make dopamine neurons more susceptible to toxins and injury. While the etiology of some of these conditions differs between species, the research into blood-brain barrier function in dogs can potentially contribute to a better understanding of human neurological diseases as well. So, to summarize, the blood-brain barrier is a highly complex and dynamic structure. It plays a critical role in protecting the brain from harmful substances while ensuring that vital nutrients and molecules get through. Dysfunction of the blood-brain barrier is involved in a wide variety of diseases, including inherited genetic mutations, inflammatory conditions, infections, aging, traumatic brain injury, and epilepsy. Understanding the intricate mechanisms of the blood-brain barrier is crucial for developing new therapies for neurological conditions in both dogs and humans. Future research in this area will likely focus on understanding how to regulate the function of the canine blood-brain barrier, especially with respect to key efflux transporters such as P-glycoprotein, to identify new strategies for drug delivery and for prevention and treatment of neurological conditions. A multidisciplinary approach that combines genetics, pharmacology, and pathology will be important to advance this field and improve the welfare of our canine companions. The dog is a valuable model for research into many neurological conditions and has the potential to significantly advance our understanding of how these conditions develop in humans. And that wraps up another episode of That Neurojar. I hope this deep dive into the canine blood-brain barrier has given you a new appreciation for this amazing system. It is critical for maintaining the health and well-being of the brain. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.